Alright, so this is a simplified setup of a long-term evolution experiment. As you can imagine, this is a cartoon, so this is meant to just explain things in a very simple yet accurate fashion. For example, there's a lot more than just glucose and citrate inside of these flasks. But for the sake of keeping things simple, this is what we're going to focus on. I don't intend to make a video that's going to take 100 years, so I'm not going to go into every single detail. If you want all of those gross details, you can read the research papers. So basically what the researchers did was they took a little bit of a parent culture of E. coli and they put it into a flask. The flask right here is labeled day 1. They just took like a drop or two and put it in here. And E. coli, under ideal conditions, are able to double about every 20 minutes. As you can imagine, when things double, they add up very, very quickly. If any of you have ever played the game before where somebody asks you, would you rather have a penny in the bank now that doubles every day for 30 days, or would you rather have $1 million in the bank right now? Anybody who's ever played this game knows the answer is you would rather have the penny in the bank that doubles every day for 30 days. Unless, of course, you got yourself mixed up with the wrong people and you really owe somebody some money, or you're just a very impatient person. So, basically, these E. coli replicate like crazy. After a few hours, there's no glucose left. So, 24 hours later, these E. coli they've already been in starvation mode for quite a while. The researchers come along, they take a few drops from here and put it into here. And again, the E. coli will replicate like crazy. To them, this is like the promised land. They will replicate and they'll take over this flask and they eat up all the food that's available to them. Interestingly, after a few hours, of course, there's no food left for them to eat. So 24 hours after this flask is inoculated, the researchers will take just a few drops from here and they'll put it over into here. And the whole process starts all over again. And this is an experiment that's been going on for over 20 years. It was started in 1988 and it's still going on today. I think this is wonderful because this is a perfect Darwinian scenario. We have a large population. Now, what do I mean by that? In this flask right here, we have more E. coli than the number of people that have ever existed on the face of this planet. So there's a lot of room for evolution to take place. There's a lot of possibility for evolution to take place. We have a very large population. That means lots of different mutations can happen. There's all kinds of different genetic changes that can happen. And we also have selective pressure. Now, what do I mean by that? Under oxic conditions, meaning when there's oxygen present, E. coli will eat glucose. And in fact, they'll eat glucose even when there's no oxygen present. Strangely enough, they won't eat citrate, which I drew as red right here, when there's oxygen present. When there is oxygen present, they don't make the transport proteins for taking the citrate into the cell. And I'll talk about that more here in just a few minutes. But basically what the researchers are hoping is they're hoping that this E. coli, or these E. coli cells, I just drew one right here, but they're hoping that the population of E. coli cells will adapt. They're hoping that there'll be some mutants that end up having an adaptive mutation that allows them to eat this citrate and use it as a food source, or a series of mutations that will adapt them to be able to use this food source. So that's what selective pressure is. It means that there's pressure from natural selection to be able to use this. Now, if you don't have the mutations in order to change the cell so that it can use citrate, then you can have the selective pressure all day and it doesn't do you any good. But these E. coli cells, when they're in these flasks and they're starving amidst abundance. Because when there's oxygen present, they're not able to use this food source, even though there's plenty of citrate around. Citrate's an abundant food source, it's readily available, but the E. coli, when there's oxygen present, they're not eating it. And the third thing is there's many generations. E. coli, when you put them in this culture, like I said, they double every 20 minutes. They replicate very, very, very quickly. They make lots and lots of copies of themselves. Human beings have a generation time of about 20 years. E. coli have a generation time of about 20 minutes. What do I mean by that? Well, human beings, they grow up, they get to sexual maturity and they're able to have children at about age 20 years. E. coli, they're able to replicate about 20 minutes. What that means is that E. coli are able to undergo the same number of generations in 2 years that would take human beings 1,000,000 years to undergo. And so that massive number of generations combined with the selective pressure and the large population really makes this the perfect Darwinian scenario. A lot of people, when they think of evolution, they think of it in terms of, oh, this takes millions and millions of years to happen. But in reality, people should be thinking of evolution in terms of generations. It's not so much about the amount of time it takes as it is the number of generations. Since E. coli are able to undergo so many generations in such a short amount of time, we should, at least hypothetically, be able to see some real evolutionary change, some really amazing adaptations. Now, let me talk... one more thing about E. coli. E. coli are very mutable. What do I mean by that? Being bacteria, they can undergo a lot of genetic changes that human beings cannot undergo. E. coli, you can do all kinds of things to their DNA, and they can survive it. But whereas a human being and a mouse, if you were to do those same types of genetic changes, it would kill them. So there's a lot more genetic changes that E. coli can have and still survive and still swim around in this environment and be as happy as they can be, even though they don't feel emotions. But they can change a lot, and it won't necessarily kill them, at least not as easily as it'll kill human beings. So there's a lot more opportunity for mutation and natural selection to come in. Alright, now let me go into the biochemistry and the genetics a little more and explain how those work. Alright, so what I'm going to do first is I'm going to draw a basic E. coli. I'm going to draw a basic E. coli cell. Now, E. coli are kind of shaped like a fish oil pill, or kind of like the little candies you can buy at the grocery store that are shaped like a fish oil pill, but I can never remember what they're called, which sounds appetizing. And E. coli also have flagella coming off their backs. And for those of you who are familiar with a bacterial flagellum, that's located right about here. That's a little motor they discovered inside of bacteria a few decades ago. I think it was first discovered in the 1970s, and it has since become an icon of the intelligent design movement. Alright, so we're also going to draw inside of the E. coli cell a bacterial chromosome. And bacterial chromosomes are round, unlike human chromosomes, which are linear. And we're also going to draw some citrate in the environment. And for some odd reason, I think of citrate as being red. So I'm going to draw that using red dots. And according to the papers I've read, there was a lot of citrate in the environment. So I'm going to draw a lot of red dots here. And there was also glucose in the environment. And I'm going to use these little blue dots to represent glucose. So here we go, we're drawing some glucose, and it's all fine and good to have these molecules in the environment, but if the cell can't use them, then it doesn't do it any good. So we're going to draw some transport proteins. Here's a basic transport protein right here, and this blue rectangle is going to represent the transport protein for bringing glucose into the cell. Now, if you've taken high school biology, they talk about cells being semi-permeable, and they kind of make it sound like cells will allow different materials in and out depending on how big they are. That's not actually how it works. I think that that's simplified to the point where it's wrong. In reality, cells have these transport proteins, and transport proteins will let in one particular molecule or a very, very specific family of molecules. You have to be just the right shape, just the right size, and just the right chemistry to be let in and out. For example, a transport protein might only let glucose in and out of the cell, and it might be one direction. It might only let the glucose into the cell, and that's it. That's all it does. It's not going to let any other molecule in and out of the cell. Some transport proteins, like I said, will let a family of molecules in and out of the cell. Others are going to be more specific than that. But all transport proteins are going to be extremely specific. They're only going to let one specific or a few specific molecules in and out of the cell. Now, let me draw another transport protein so that we can bring the citrate into the cell. Now I'm going to draw this as a different shape, just to get you the idea that not all transport proteins are identical. They can be different shapes, different sizes. Actually, I think that's ugly. Let me redraw that. Here we go. We have our transport protein for bringing citrate in and out of the cell. So this transport protein will let citrate come in. Now the problem is that when there is oxygen present, this particular transport protein is not made, which is kind of interesting. So when there is oxygen present, this citrate's going to come along and it's just going to bounce right off the cell. You can kind of think of these like pool balls on a pool table, all bouncing off of each other, bouncing off the cell, all in a 3D watery environment. So unfortunately for the cell, this citrate comes along and it just bounces right off. So it's able to use all of these glucose molecules, but then these citrate molecules are just sitting there. This is a great food source for the cell if it can import it. Unfortunately for the E. coli, in this particular situation, in this particular environment that the experiment was done, the E. coli cells were starving amidst abundance. So there was all this food out there, but because they weren't making the transport protein, this wasn't being brought into the cell. And I'm going to explain the genetics and the regulatory information of I'm going to explain how the regulation, how the genetics of this works in just a second. But for now, this is basically how it works. The transport proteins will bring glucose. This transport protein will bring glucose into the cell, and this transport protein is not being made because there's oxygen present. And of course, there's plenty of other transport proteins all around the cell, bringing dozens and dozens at least of different molecules in and out of the cell, and maybe even more than that. Alright, so what we have here is we basically have our bacterial chromosome. And on this chromosome, you're going to have hundreds and hundreds of genes. But I decided to represent just one as a blue rectangle. In reality, you won't have it. Genes are not a rectangle sticking out from the chromosome. But obviously, I need to make this simple in order to make a video that doesn't take 100 years to watch. So what we have right here is we have our gene, and next to our gene we have our regulatory information. Now, I also drew it over here. And I drew the gene in blue because basically genes are blueprints that code for a protein. So right here, this blueprint, this gene, describes what this protein should look like. And if this gene is off by even a little bit in the wrong spot, this protein is not going to be made properly. Now, there's a little bit of leeway, of course, but I'm not going to go into all that. But for the most part, this gene has to be just right in order for this protein to be made. Now, over here we have our regulatory information. You can kind of think of this like an on-off switch. But in reality, just understand this is a lot more sophisticated than that. A better analogy than an on-off switch would probably be an artificial intelligence system. This regulatory information not only tells you when it's appropriate to turn this gene on and when it's appropriate to turn it off, it also tells you how much of the protein should be made. It is able to figure out, based on talking to other parts of the cell, what's going on in the cell. And by figuring out what else is going on in the cell, it can say, okay, we need 100 of these proteins, or we need 1,000 of these proteins, or just turn the system off entirely, or turn the system on and just keep on making proteins. Regulatory regions are very complex, and they're a lot more complicated than an on-off switch. But for our purposes, we're just going to think of this as an on-off switch. So when there's no oxygen present, this information is turned on. So when there's no oxygen present, the cell is going to read this region of DNA. It's going to read this gene, and this protein is going to be made. But when there is oxygen present, this is going to be turned off. So this right here is not going to be read, and this protein is not going to be made. Simple enough, I think. Alright, so a couple years into this experiment, the researchers ended up discovering an amazing adaptation. Rather than the citrate coming along and just bouncing off the cell membrane like it would normally do, the citrate instead was able to enter inside of the cell membrane. Now obviously it wasn't passing through the membrane itself, it was passing through some kind of transport protein. But the researchers weren't sure what was going on. They weren't sure what the molecular basis of this was, they weren't sure what the biochemical basis of this was. But basically, somehow, the E. coli had adapted in order to eat the citrate, in order to use this as a food source. Now, in a paper that they published in 2008, they said, "The new CIT+ function has been the most profound adaptation observed during the LTEE, and has had major consequences." In other words, according to these researchers, the long-term evolution experiment has seen a lot of different adaptations, it's seen a lot of changes, but this one has been the most profound. This is the most amazing change, the most amazing adaptation they've seen over the course of this experiment. Jerry Coyne, who is a famous evolutionary geneticist, Richard Dawkins views him as being the top evolutionary geneticist on the planet. He commented that this is a poke in the eye for anti-evolutionists. And anybody who's followed my channel before, I've commented on Jerry Coyne's videos in the past. However, nobody knew what was going on at the molecular level. Basically, all we knew was that there was an adaptation of some sort, some kind of biological change happened that allowed the citrate to enter inside the cell. And why was that? It wasn't, nobody knew. It wasn't until 2012 that a paper was published where we actually were able to figure out exactly what was going on. Now, right off the bat, there are several different possibilities for how this adaptation might have come about. Perhaps this regulatory information got corrupted somehow. So perhaps this little on-off switch isn't working properly, and so now this protein is being produced even though it's not supposed to. Something went wrong with the regulatory information, and now we've got this protein being produced perhaps even all the time, which is great for the cell because if we have this protein being produced all the time, then at least under this bizarre scenario, we have it where the citrate is being brought into the cell, and the cell is able to use this and metabolize it and go on and produce more cells. So it's no longer starving amidst abundance. Now, I think that is the most likely scenario. However, in 2008, when the paper that I read a quote from was written, nobody knew what was going on. So this was just one possibility. A few other possibilities were these. Perhaps there was some other transport protein, and I'm going to draw a green triangle to represent the next transport protein, and perhaps this transport protein was designed to bring some other molecule into the cell besides citrate. Now, every once in a while, this sort of thing might happen, where you have a transport protein that's coded for by the genetic material, and this transport protein gets corrupted in some way. So it's no longer as specific as it's meant to be. And because it's corrupted, because it's deformed basically, it's able to accept citrate into the cell as well. Even though it wasn't designed to, even though it's not supposed to. Now this is great because now this cell is able to take in the citrate. Granted, it's because there's a defective protein, but the cell doesn't care. It thinks this is great. It's able to metabolize this citrate now and able to survive and reproduce and make lots and lots of copies of itself. It can make a lot more copies of itself than its brothers and sisters can who don't have this mutation. So just because something is a beneficial mutation doesn't necessarily mean it's an onward and upward evolutionary change. Very often things like losing your eyes in a cave can be adaptive. It can be very beneficial. Very often natural selection favors losing some trait that things had before. And in this case, it's possible that because this protein was corrupted, because it's deformed now and not being made properly, it's very possible that perhaps the cell is using this as a way to bring citrate in. Now, there's another possibility as well, and basically, perhaps the cell has developed some fundamentally new biochemical pathway. Perhaps it's able to develop something brand new, so it's able to bring citrate into the cell or metabolize it even outside the cell. Perhaps some fundamentally new thing was developed. Perhaps the cell took one of these other transport proteins and duplicated it, and then as a result, it was able to use the or duplicated their genes. Let me say that properly. Perhaps there was a duplicated gene from some previous protein, and perhaps this new duplicate was able to be mutated and changed and molded by natural selection in order to become some brand new wonderful thing that's able to digest the citrate and able to metabolize it in some wonderful way we've never seen before. Or perhaps this is a new protein that was constructed using previously existing genes that were duplicated, and perhaps this has developed and been molded by mutation natural selection just so that it brings the citrate into the cell. Lots of different possibilities. But bottom line, with evolution, every now and then we should see mutation and natural selection lead to something brand new, some fundamentally profound and wonderful biochemical change, some brand new biochemical structure, some brand new cellular pathways, something new and wonderful that we've never seen before, with plenty of biochemical sophistication to it, that we can watch being constructed in the laboratory. Perhaps something like this was made. Now, since I know the answer, I know what was going on, let me explain to you exactly what happened. Alright, so basically what we had is, we have our original setup right here. We have our gene, our blueprint for our protein right here, and we also have the regulatory information. Now, what happened in this particular scenario is, this gene right here got duplicated. So there ended up being two copies right next to each other. And this right here was actually a beneficial mutation. But the researchers didn't catch it at first because this right here only gave the cell an increased fitness level by about 1%. So it only gave it about a 1% increase in its ability to survive and reproduce. Now, later on what happened is, this tandem duplication ended up getting moved around. It ended up being taken from this part of the genome and ended up being moved next to a different on-off switch entirely. Now, this on-off switch was on even though there was oxygen present. So either this on-off switch is turned on in the presence of oxygen, or it just doesn't care that the oxygen is there. Whatever the case may be, this particular on-off switch was on under the environmental conditions that the E. coli were grown under. Now this is great because now the E. coli have two genes that are actively making that protein, that transport protein that I mentioned earlier. Now, let's look at Jerry Coyne's claim that this is really a poke in the eye for anti-evolutionists. That's really not an accurate claim, especially since the time when he made this, nobody knew what was going on at the molecular level to begin with. It was really a premature claim to make. A lot of people have accused creationists of denying that species changed over time. Very often, if you go to high school biology classes, or if you sit through the evolutionary biology class that I sat through, they'll tell you that creationists deny that species change. They'll tell you that creationists deny natural selection. They'll tell you all of these things. But anybody who's ever read Origin of Species will know that even in Darwin's time, people knew that species changed. It wasn't a secret. It wasn't something that people debated. They did debate, however, how much change was possible and what was the basis of these changes. So because people didn't understand genetics in Darwin's time, there was a little bit of debate about, there was actually a lot of bit of debate about natural selection because it was a popular subject. However, it wasn't originally Darwin that came up with the idea. It was actually a creationist by the name of Edward Blyth who first came up with the idea of natural selection. So mutation and natural selection are accepted by creationists. Creationists have always accepted that species change. I've gone into that more in my other videos before. Intelligent design advocates, of course, accept that species change. They accept natural selection. The debate between creationists and evolutionists and intelligent design advocates is not whether or not species can change. It's whether or not these profound adaptations, like taking scales and developing those into feathers, or taking arms and developing those into wings, it's whether or not these profound adaptations, these grandiose claims that neo-Darwinists say that mutation and natural selection is capable of, are really possible. Is mutation and natural selection, or are mutation and natural selection really capable of taking a simple prokaryote that lived in the ocean billions of years ago, a simple one-celled thing swimming around in the ocean? Can mutation and natural selection take that and develop all of the life that we see on Earth today? Is the evolutionary tree of life really grown from mutation and natural selection? Can you take fish and develop philosophers? That's the type of thing that the debate is about. It's not about whether or not things change in general. It's about what types of changes are possible and what types of changes did happen. And in this case, even though this, according to the author, was the most profound adaptation they've seen over the course of their experiment, in the context of creation versus evolution, this is definitely not a poke in the eye for creationists. Everybody had pretty much agreed right along that this sort of thing can happen. We haven't seen the gain of any new genetic information. All we've seen is the duplication and the rearrangement of genetic information that was already there. So it's basically like we took a lamp and we duplicated it, and then we plugged those two lamps into a completely different outlet. And that's a terrible analogy, but whatever. All we've seen here is a duplication of pre-existing genetic information and a rearrangement of that information. We haven't seen the gain of any brand new genetic information. Now, one person I talked to said that, perhaps these duplicated genes could be used to go on to develop new biochemical pathways, those brand new profound evolutionary changes that neo-Darwinists say are possible given mutation and natural selection. And you know what? That's always a possibility. Unfortunately, that's a scenario that is basically unfalsifiable. In other words, it can't be experimentally disproven. At least not to any extent that I think that neo-Darwinists would accept it. However, not only are creationists criticizing neo-Darwinism, saying, hey, look, are mutation and natural selection really capable of making these profound changes that you say that they can make? There's also scientists who are secular, who are even evolutionists, who criticize neo-Darwinism, saying, you know what, I really don't think that mutation and natural selection can produce those profound types of changes that you say that they're capable of producing. And the reason is, whenever we look at mutations in the laboratory and we understand the molecular basis, they're very interesting to us as biologists, but they really don't show that natural selection is capable of performing. It's not able to live up to its claims. It's not able to live up to the promise that neo-Darwinists make. They've never been shown to do this. So that's basically what the problem is, what the debate is about. In this scenario, once again, all we saw was a duplication of pre-existing information, and we saw a rearrangement of the DNA so that different parts of genetic information ended up next to different parts. We saw some genetic information end up next to some other piece of genetic information. We didn't see the formation of anything fundamentally new. We haven't seen the origin of new genetic information. And you know what? If neo-Darwinists want to, by all means, do more experiments and show how these profound changes, like fundamentally new biochemical pathways, can come about. Keep going with E. coli, keep going with experiments like this, and see what we can produce. In fact, intelligent design advocates and creationists perform these types of experiments as well. But this, I think, is really getting close to what a term that Michael Behe coined. He uses a term called the edge of evolution. And what he means by that is he thinks that mutation and natural selection have limits in what they're capable of. There's this outer limit of what mutation and natural selection can do for any given organism. So basically, I think this is kind of approaching that. And other people have commented, too, that they think this is really approaching what Michael Behe calls the edge of evolution. And of course, as a creationist, I object to just labeling every single genetic change that happens as evolution, because you're lumping several completely different ideas together and pretending they're all one idea. Just because species can change, and just because one species can split into two species, which everybody accepts, and just because natural selection happens, which everybody accepts, does not prove that mutation and natural selection are capable of taking a fish and turning it into a philosopher, for example. There's definitely... all of the evidence is really showing that there's definitely limits on what biological change can and cannot do. So that's what I have to say about this. No new genetic information. All it is a duplication and a rearrangement of pre-existing information. Might genes like this go on to develop into something completely new and something completely profound, or some fundamentally new biochemical pathway? It's possible. But neo-Darwinists really haven't presented the evidence to show that those types of profound evolutionary changes can happen. They've shown that mutation happens, they've shown that natural selection happens, and I think that they have a very... even if you accept the universal tree of life, they haven't shown that mutation and natural selection are sufficient to explain the universal tree of life. I don't accept it. However, Michael Behe, for example, does accept the universal tree of life, but he thinks that intelligence was required in order to make those profound, those fundamental biological changes happen, even though he thinks that human beings, for example, are related to fish. Alright, so I hope that that was a decent explanation. I hope that everybody understands what's going on here, and I hope that everybody enjoyed this video as long as it was. Check out my website. Anyway, you'll see my two websites come up here in a minute. I've talked about them in previous videos, but this video has gone on long enough, so I'm not going to describe them right now. Go check out my other websites, read more about this experiment, and see what you think. Don't just take my word for it. This is Greg, out.